Mitosis and Meiosis, a full explanation by Leo Maldonado and William McClure. Mitosis, the division of a single nuclei into two genetically identical nuclei. Prophase, step 1 of mitosis. During prophase, the chromatid fibers begin to coil into two identical sister chromatids, and mitotic spindle begins to form. The mitotic spindle is attached to the two centrosomes. Late in prophase, the nuclear envelope breaks up. The spindle attaches to the centromere and pulls the chromosomes to the center of the cell. Then metaphase begins. Metaphase, step 2 of mitosis. Now the chromosomes are aligned in the center of the cell and are held firmly in place by the centrosomes on either side of the cell. Mitotic spindle is also fully formed. Anaphase, step 3 of mitosis. Anaphase begins immediately once the sister chromatids split, now becoming full and identical daughter chromosomes. Chromosomes move towards opposite ends of the cell as the mitotic spindle shortens. Simultaneously, the spindle not attached to the chromosomes splits up, elongating the cell. Telophase, step 4 of mitosis. This phase begins when the chromosomes have reached opposite ends of the cell and nuclear envelopes begin to form again. The spindle disappears, chromosomes uncoil, and the cells separate, thanks to the help of cytokinesis. Why do cells use mitosis? Cells use mitosis for easy and fast replacement, growth, and development of cells and the body. Go.
Meiosis, a two-phase process that ends up with four genetically different cells. Meiosis 1, prophase 1. As the chromosomes coil up, special proteins cause the homologous chromosomes to stick together in pairs. This crossing over causes a rearrangement of genetic information. As prophase 1 begins to finish, the spindle forms, chromosomes coil up more, and are moved towards the center of the cell. Metaphase 1. The homologous chromosomes are aligned in the middle of the cell and are held in place by the centrosomes. Anaphase 1. The attachment that held the homologous pairs together breaks, and the chromosomes move towards opposite ends of the cell. It is important to note that the sister chromatids migrate together and don't split up. Telophase 1. The chromosomes at opposite ends of the cell. Each pole now has a haploid chromosome set, although each is still in duplicate form. Cytokinesis occurs and two haploid daughter cells are formed. Meiosis 2. The process of meiosis 2 is the same as mitosis, with the exception of the haploid cell not undergoing chromosome duplication. It is important to note that these changes are occurring in both the cells produced by meiosis 1. At the end of meiosis 2, there are four genetically different cells. Why meiosis? Meiosis is a form of cellular reproduction that causes genetic variation. This allows for organisms to adapt to their environment and change as is needed. Differences between mitosis and meiosis. Meiosis produces haploid daughter cells in an organism. It has half the chromosomes as mitosis and exchanges genetic material. When meiosis is completed, there are four cells. In mitosis, there are double the chromosomes, and the end result is two genetically identical daughter nuclei. There is no exchange of DNA.